Okay, let's get started. And then, again, I apologize for the stream cutting out last week. So, did you all see the announcement at least where I linked to last year's lecture so you could see that content again since, like I said, it completely cut out. There was no way to save it at that point. And then just a reminder that we have our final presentations today. Sorry, I didn't map my Wacom yet either. Come on. What is this doing? Oh, sorry, wrong screen. So, final poster presentations. And I have received many emails. So I printed out a few slipped by. And then, if you haven't already heard, the toner is down on the first floor, which it is like never down, but obviously it's down the day that we need to all print. But they should be in soon. I might go check like during our break and try to print out the posters, and hopefully they'll be in. And so again, that's going to be our lab today. Is the poster session. And again, hopefully everybody got the memo that we're trying to combine Tuesday and Thursday today. I know a few people have already said that you have work, so you couldn't make it, which is fine. So we'll do a small makeup group on Thursday session during the lab time. So if you're Tuesday and you're sick today, last chance is Thursday then for poster presentations. Unless, of course, you make like individual appointments, but so reach out to me, right, if for some reason you can't make it out to Tuesday or Thursday this week. And then we're basically at the home stretch here. So we're going to finish up this week's lecture. We're looking at kind of polymers and at least just getting a little bit of an introduction to that. And then next week we'll do a review on Tuesday like we normally do, and then test Thursday officially. And then a few people have already reached out about taking the exam early, so I can have that ready to go to the testing center now. Other questions, comments? Okay, so we will revisit the printing thing. Like I said, during break, we'll try to see how it's doing. All right, so let's look at our last polymer chapters. And then, did I post the new link to the inner chapters? Oh, I had to like upload them all, right? So those worked fine, all the uploaded files on Blackboard. Uh-oh. Oh, but you mean like lectures. Okay, but all the inner chapters themselves are posted. Okay, yeah, I'll hopefully fix the link. Let's try to do that. Okay, I'll try to work on that. All right, but just as long as all the inner chapters are at least posted, and then I'll try to get the links going. All right, so now we're looking at polymers, and we'll do kind of a cool demo. And so here are some examples of polymers. DNA is often given as kind of a natural polymer. Nylon is another polymer, or other like synthetic fabrics. And so, well, what is polymerization? And so polymerization is just defined as our repeated addition of small molecules to form a long continuous chain. And so you can take a whole class on polymerization and how to control polymerization, really. And so some examples is maybe you could be doing just with one monomer. And so, well, let's define what a monomer is. And so a monomer is just a small molecule or unit from which polymers are synthesized. So we could be working with one monomer and just make this AAA chain, for instance. Or you could have two polymers and maybe make some sort of, alternating chain there. So those are kind of the common ones, at least to control. Obviously, it'd be harder to control like other patterns, but again, that's what kind of the research and reaction conditions are for. Here's another just a representation, right? We have the monomer of our single unit of maybe this chain length, and then, right, the whole chain. Another good analogy that people usually bring up, so right here's our unit, so our monomer, polymer. And so if you're teaching this to like children or something, there's always a train car analogy as well. So you often see this where like the individual train car is again the monomer, and then the whole train, which I'm going to draw a horrible train. That was a poor attempt at an engine. Yeah. Some weird, like, it almost looks, yeah, no, cat dog variation, no. But then our train is our whole polymer. So just other ways to kind of think about it. And so then kind of looking at a few mechanisms. So this is one mechanism. So often polymerization occurs by a radical mechanism. And so there's often a lot of research too devoted into radical mechanisms. Try to pull up some papers. That's kind of like where the current hot area of focus is like kind of these, especially UV initiated reactions. So maybe you have some sort of catalyst. And like I said, I'll try to find some example real quick while we're talking. But some sort of UV initiated catalyst, and then this kicks off a radical mechanism. And so sometimes, I think we've looked at it. So sometimes with chlorine, like we've seen this with chlorine, right, where that bond splits to create the chloride radical. And so again, that's often UV initiated. So similar mechanism for our ethylene polymerization reaction here. You mean here? Or? Yes. Oh, this is a double bond. Oh, okay. Yeah, but I'll rewrite it. So to your point, so let's see this mechanism in a little bit more detail. So I think you did this in lab, right? And yeah, that does look like an equal sign. They probably did use an equal sign as their double bond from wherever I stole that text. And so, right, again, just kind of putting your brain in the correct geometry frame of mind. So this is planar, and then we have our radical comes, and then we can make a bond. And so these electrons, right, go into a bond here from our kind of double bond. And so then looking at our product, we have our, whatever our radical was, so they have this hydroxide radical mechanism as the initiator. And then so CH, H. Now we have, right, this is a single bond. And then this other electron is now a radical. So that's what's shown up here, right? This process, like I said, maybe not the clearest representation there. But then you can see how it goes on, right? So there's our radical initiator. Now we have our product, right? But then our product has a radical. And so this could potentially continue on and on. Like I said, this starts getting into kind of research conditions and how to control your length of your resulting polymer. But then, for instance, so then, right, this radical will go on to react with another double bond. Again, how you did it in lab last week. So then here we go again, bond, right? But then we're left with a radical again. Oops, sorry, that should have been a radical. Radical. All right, so now we have essentially our two monomers put together. And so that's something that you want to be able to do, right, is like identify, well, which monomers came together. And okay, so we're looking at our monomers, and again, right, radical. So this can go on and on and on in a radical mechanism. And so then how do we get to termination? Like, when are we done? And again, right, you would get into more specific conditions, reaction conditions, concentration, heating times, reaction times, all that good thing, right, to kind of figure out how you can control this chain length. And so, so that's where the research side of polymerization comes into, because basically you're kind of trying to control the reaction mixture to a certain size, and then, well, what causes it to terminate, right? And so basically, we have a reaction termination when it just so happens that our radicals meet, right? So again, this is what contributes to chain length, and that's where you would get into exactly how to control it, start testing different conditions. But I'll just try to simplify this a little bit. So then you have your chain come along, and then if it meets, right, with another radical, so now our two kind of polymers, right, and if I was doing this, probably if I took a little more time, we could abbreviate it, right, with this N chain. So N, how many monomers we have repeating. And so then we can take our polymer, so polymer, polymer, and then again in this reaction mixture, eventually they meet and then are terminated, right? So then your two radicals meet. Now there's no more way to create a radical, and so this is again what results in termination. And so kind of simplified there. Sorry, this is a H up here. All right. So there's at least the basics of how polymerization occurs. And I mean, really good area too to get into of like polymers, right? So that's like literally everything is plastics, manufacturing, and petroleum, right? Taking petroleum derivatives and converting them to plastics. And so then we can just start to appreciate like why plastics have different names and different properties, right? It's all going back to whatever monomer. And so of course, right, you may be familiar with Teflon. So Teflon is used as a non-stick coating. That being said, I would not use Teflon personally. These things tend to break down. Yeah, I mean, I would think that fluorine's reactive, so I'm surprised that like Teflon's robust in the first place. But I mean, you can like scrape it off, so I don't trust it at all. I cook with cast iron, for the record. Okay, I'm trying to find just some other. Yeah. Yeah. Kind of typically speaking, yes. I mean, uh, yeah, I mean, that's going to be like for all these monomers, like with plastics, because we could get into like styrene, but we'll also look at like DNA as polymers, and then it might look like slightly different, but we'll look at how to shorthand it. So yeah, any sort of, I mean, I'm not planning on getting too deep into this. Actually, I have a nice poster where I did like an outreach thing about the different plastics, but I mean, it's all just down to the monomer, and then it explains like why things are difficult to recycle. Like for example, going back to styrene, actually, why it's a pain is because of this group here that it does. It no other plastics have that. So then it's like, well, how do you recycle it? And then it goes into the numbering system, which yeah, we could go down that rabbit hole, but okay, I was trying to pull up some papers, but well, I don't know that these are polymer reactions, but at least you can see again like what people are doing for research, that they have testing different LED conditions, right? Just trying to functionalize. And so that's another big thing, a high area of research is like trying to functionalize these CH bonds. Obviously, they're pretty inert. So then trying to use any sort of catalyst. And then you can see, right, same sort of, this is probably a radical mechanism, I would assume, since you're doing a similar transformation, right? So you're taking your double bond and creating a new bond there. So anyways, we won't go down. There's my printout. And so then, like I said, we don't want to get too deep into it, but just at least appreciate that this is what dictates, right, all the different numbers. So I don't have that like handy with this chart. Like I said, I have that outreach poster. So we looked at ethylene, and usually you have like high density versus low density polyethylene, and that can go into like branching. So high density, they tend to not have a lot of branching, and then they pack super tightly, versus low density, they tend to be more branched, and then that gives it, again, different kind of properties. Polypropylene, so notice, right, it has that little extra methyl group on the end, and again, just kind of dictating the properties, so used in like pipes, and then you're probably aware uh, the polypropylene can be a little bit harder. Same thing, so PVC, right, it has this vinyl chloride group, although, right, you could also start to wonder like what kind of off-gassing and weird organic things it is outputting. So that's like the PVC material, of course, more pipes and tubing. Are y'all familiar with neoprene at all? So yeah, that's the alternative. to nitrile. So if you did have a nitrile allergy, you could use neoprene. And then methyl methacrylate. So I'm pretty sure that this was the chemical that spilled the other day. Did y'all hear that on the news? Yeah, this was in like Southern California. They had some plant was leaking out methyl methacrylate. Not really sure what they were doing. And then, I mean, not to go down another rabbit hole, but that always gets into like issues of quantity and like how much more different and dangerous like trying to engineer a process is. And like methyl methacrylate is not dangerous, right, if you're a little bottle of it. But now when you have like, it was obviously pressurized or something, right? Like the container was pressurized and heating, and who knows like exactly what was going on in their process. But I think that just kind of shows you how chemicals can kind of get out of control once we look at them on a large scale. And I assume they must have been manufacturing, you know, some of these things. So obviously, this is a monomer for plexiglass, and so I mean, just look around, right? There's a lot of clear glass everywhere, so I'm sure they were using that. that. Do you use it in like your office? Oh wow, that's that's always interesting. I always wonder like how they really developed that. Like just 'cause there's an extra step, you know, of like obviously there's like, you know, a chemist tests it, but then now you have to test it for like extra stability in the body and how it reacts and like. Oh man, well, that's also kind of crazy. And so yeah, there's Teflon again. And then I'm not too familiar with this one, like where you would find it, but apparently it's in a lot of like tents. So I don't know if you camping people are familiar with acrolein or. So this is acrylonitrile. Nitrile. Sorry, can't pronounce that one. But you can see it has that nitrile group on there. And there's polystyrene once again, with again this styrene group that makes it pretty much a nightmare to recycle, unfortunately. Oh, kind of good news while we're thinking on this topic. And so I think some people did their poster on this, which should be interesting. So, of course, microplastics is a big topic of discussion. And so let's kind of talk about why. And so, right, we're looking at all the polymers, but what we didn't say is, okay, well, now that we've made these polymers, right, we've made these incredibly strong bonds for the most part. And so now that's kind of the problem that the chemists are encountering is like, well, great, we made plastic. How do we break it down? And so obviously, right, there's research into bioplastics. And then so part of the concern or where like microplastics comes into is once you have this monomer, it's hard, or excuse me, once you have the polymer, it's hard to break it down right back into the original monomer. And so the fear is that it just kind of physically degrades right into smaller and smaller plastic parts, but doesn't actually chemically degrade. And so that's where, right, people were researching microplastics and finding microplastics in fish and on Mount Everest and I don't know, in the air and everywhere. But at least some bit of perhaps good news on this. Is so now they're saying that maybe they've overestimated the amount of microplastics. I mean, obviously, right, there's still probably microplastics, but they were saying that there was some confounding evidence. So basically, that the nitrile and latex gloves that the scientists were wearing when they're doing these measurements is apparently shedding and perhaps contaminating the samples themselves and leading to higher elevated levels. And so I was hoping well, I was looking for the original study, but I think basically they had like compared two different types of gloves. So apparently they're like special made gloves that like do not shed or like do not shed as much as our regular like lab gloves. And so they kind of compared that, and we're comparing kind of the contamination. So yeah, they tested several different types of gloves, including clean room gloves, and found that these gloves gave again higher incidence of false positives. So I mean, right, just something to think about. I think that's really important as a scientist, that you try to stay as objective as possible. And that's obviously like really hard because you want to find something, and you could have in your mind how things work or what's going on, and then you just want the data to prove it. But you have to be, again, kind of really careful that you're looking at all the factors and everything. It's kind of very easy to be swept away with your own kind of insular thinking and think you're correct. But anyway, so there's that. So you can continue doing research on that and figuring out how to break down these monomers, and I'm sure they're developing new biomonomers and polymers by the day. All right, so let's look at our next type of polymerization. So we mentioned that there are kind of two main mechanisms, and so the other mechanisms for a polymerization reaction was the condensation mechanism that we'd mentioned. And so we're once again losing water to form a bond. And so now we have this carboxylic acid reacting with our amine here. And then this right here is our amide bond formation. And then again, right, no water. And again, you're kind of thinking in the back of your mind about potentially this Dean-Stark reaction. So I know we've mentioned it, but we'll bring it up again, right? Just so you're kind of thinking, okay, physically, we have to figure out, right, here's a similar reaction. So again, you can also do it with an alcohol, similar concept. But again, right, how are you going to get off that water? So we mentioned our Dean-Stark apparatus, our solvent with our lower density, or higher density than water, or lower, sorry, I don't know what I'm saying. A solvent that is less dense than water, that makes more sense. And so water goes on the bottom, and then we can drain it out. And so let's kind of look at this mechanism again. So our basic mechanism. So we have our carboxylic acid group. And so then we have our amine, which is our base, right? And so now our base can attack. And so our carbon here would be what's called electrophilic. Again, we have this double bond, so that's kind of pulling electron density away from the carbon. And so electrons initiate the action, they attack this carbon, and then right, we have this double bond for the electrons to go up. And so now electrons are on the oxygen stabilized. Oops, sorry. So then next steps, now we're just kind of drawing out what that looks like. So OH here, we have our O, negative charge on that, nitrogen, HHR. And so this is again what I would call the proton transfer mechanism, or some sort of acid-base mechanism. So I'll kind of call it proton transfer, or, right, we need some sort of formally base deprotonating this since it has a positive charge. And then you'll have HBA, and then this plucks this back up. So like I said, I'll let your OChem professor be nitpicky about this protonation, deprotonation, proton transfer step. So I'm just going to call it proton transfer or protonation, deprotonation. And so we're trying to form our leaving group, which is water, right? That's all we're essentially doing. So negative charge on the oxygen still. So 1 hydrogen left on our, well, amide now that's forming. And so, okay, so we have our leaving group. Now we have this, right, oxygen with the negative charge still. So this can go ahead and formally reform this double bond. And then in doing so, now we have our leaving group that can leave. So water is a good leaving group. And so that leaves us back to our product here. So again, water left, and then the amide bond formation there. Make sure everybody copies that down for a second. And then, so I'll bring this demo for Thursday, the nylon synthesis. But this is a pretty fun one as well. But so it looks like we have it drawn down here. So let's go through it. Again, similar mechanism, right? And so you can kind of start to see what the main takeaway points are. And so we have, right, these two amines on both ends. And okay, let's get into our naming too. And so how we do this nylon synthesis, at least like I said, the one that I'm going to do, there's obviously different variations, and you can start to figure out where. And so the specific one that I'm going to do with these two chemicals in particular is called nylon 6,6. And so it's called 6,6 for the 6-carbon chain between them. And so we're going to be reacting 1,6-diaminohexane. So, okay, right, we have 1, 2, 3, 4, 5, 6. So hexane, right, there's our 6-carbon chain there. Diamino, so we have an amine on both sides, right? So we can pair with our carboxylic acid, and you're kind of mentally seeing how you're going to pair your monomer. And so this is also essentially, right, like an AB monomer to polymer. Sorry, cannot talk today. And so, right, that's how we're going to get our repeating patterns from having the amines on one group, and then they'll be, again, reacting with the carboxylic acids of our other. And so our other reagent is hexane dioic acid, or adipic acid. And so we can see it has 1, 2, 3, 4, 5, 6 carbons on it again. And so let's sketch out at least the mechanism briefly, so we can at least kind of see how this is formed. So I'm trying to abbreviate. Well, actually, that's not a very good way to abbreviate. So properly, right, you could kind of do this, and then there's 4 of them, so then that helps. All right, that's more clear. I just get tired of drawing our chain out so many times. And then, again, same thing, right? We're going to see how it reacts. We have our amine once again. I'm just kind of trying to abbreviate a little. So CH2, there's 6 of them. And then an amine. And so just start putting this polymer in your mind, right? So you, okay, we're going to make a bond here, right? This is going to leave, our hydrogen's going to leave. We don't need to necessarily go through the mechanism again. But then you have another end, right, where again it can do the same thing with another carboxylic acid. So I'm just kind of trying to abbreviate now. So same thing, it can attack. And so now this is kind of, right, our repeating chain here. So this is, right, kind of our AB dimer now that is repeated, right? So again, you have your hexene diamine, so that's one part, and then it reacts with your carboxylic acid, and then it keeps chaining over and over, right? And you can tell from the way it's drawn, right next you have another amine again. And so however you need to draw that out, so I can kind of, right, abbreviate it as well. So we said, okay, we had our carboxylic acid, we made our amide bond, and CH2, right, 6 of them, amide bond again, okay, so you can kind of see, right, how it repeats over and over again. And again, be able to identify amide bond formation, right? So you can pluck out the original polymers as well. So A, B, right? A, B, kind of, etc. And so we could also refer to this as a copolymer. That's just a polymer, again, made from one type of monomer here. So that would be our copolymer. All right, so then a very similar but slightly different is these polyester reactions, which we can compare back to our esterification reactions. So if you vaguely remember from last chapter, right, these esterification reactions were where we took a carboxylic acid and an alcohol, and then you made the esters, right, which we said were fruity smelling. I don't know, I just put happy smelling. But basically, we can have the same sort of now, right, polymerization reaction. And so again, it's officially called an esterification reaction, but right, this is basically another condensation reaction, which is a different name, right? And so instead of doing the amine and the carboxylic acid, now we're doing the alcohol and the carboxylic acid there. And so it's just like we said, our ester synthesis. And so let's just look at that mechanism again so we can compare the similarity and see that again. It's also just good kind of practice for OCHEM since that's all you'll do. And so, right, again, similar sort of mechanism. It's just now we have an oxygen that's primed to attack instead of our amine. And then, right, again, action starts at the electrons. This is also hopefully going to be a repeating pattern in OCHEM that you'll do a bazillion times. But this carbon is always susceptible to attack, so we would again call this a nucleophilic, or excuse me, electrophilic carbon, since it needs electrons. And right, your hint is this double bond. And so again, similar mechanism, right? Our oxygen takes those extra electrons and takes that negative charge. I'm just redrawing the rest of it. And then we have our new bond forming. So now this has a positive charge on our alcohol. And then, right, we can see that it's going to be a similar mechanism, again, with the idea being that we want to create a leaving group, aka water. And so again, similar sort of proton transfer and deprotonation, protonation mechanism. We'll leave that for the experts. So proton transfer slash protonation, deprotonation, and so I'm just going to essentially do a proton transfer, but like I'm saying, you're going to need a true mechanistic study and probably the opinion of your OCHEM professor to really determine what it is. So that's where you can get into isotopic labeling experiments, but that is not for this class. So there's water, and then we're almost there, right? So same sort of action. So we're going to reform the double bond that causes our water to leave. So there's our double bond reformed, and then, right, the alcohol attached there. And then, of course, right, when we did it earlier with our last chapter, right, with the carboxylic acid and the alcohol, well, we only had one end, so it didn't keep going. But notice for this polyester, right, the polyester is going to keep going since we have the alcohol on both ends in this case and the carboxylic acid on both ends, right? So just kind of pay attention to that as well. So that'll probably be on the exam, is like identifying the correct monomers. Like you could be shown a polymer and then you'll be asked, right, what are the monomers? And so just keep in mind of doing this reaction, right? So you have to have, like we're looking at either carboxylic acids on both sides or alcohols or maybe amines on both sides, right, to keep that chain reaction going and going. And so this would be another example, right, of our kind of copolymers. So, right, again, this can be called A, this can be called B, and then they can make this AB repeating unit over and over again. And then again, we could define this then as our copolymer of our two monomers that come together to repeat. All right, we're kind of flying. Well, there's a lot in here, but trying to see how far we get. So before we take our break, let's at least just kind of look. Well, okay, your book is old. I admit that. So these stats are now probably 10, 15 years old at this point. But again, all we're appreciating is, well, a lot of petroleum products go into making all these plastics, and I believe it's about 50% of the petroleum, right, goes into plastic making. And so um, if you wanted to at least have a debate about fossil fuels or something, you obviously have to account for that most of it goes into plastic production as well. And then again, all we're really saying is like everybody uses a bunch of plastics. I don't know how accurate these stats are. Obviously, kind of the newer papers or sensationalized news articles that come out are usually that like the Asian countries or like China, India, they tend to like produce more plastic waste or even just have like more packaging. I mean, obviously, I think everybody's pretty guilty of that worldwide of using a ton of small packaging. And I mean, those are large manufacturing questions as well. And I mean, there's also manufacturing trends, and people go through different uh, product lines or even just switching, right? Like we went from paper bags to plastic, then those thicker plastic. Now we don't get bags in Oregon. But I mean, there's all sorts of physical concerns too. And I mean, not to get into like the politics or anything of it, but you know, really behind the scenes, right? There's all these brokers too managing all that oil. Now I forgot, like there's probably a cool movie or something about it, right? But that's kind of like who does all the deals too, is all these like brokers, and then they relabel the oil as from different countries as politically necessary. But I mean, basically we all use oil and we all need it, so we all fight over it. In both plastics and obviously moving vehicles. All right, let's take our 10-minute break slash I might run a little late, so I was going to dash down to the first floor printer and see how it is doing, and then continue to print out the fat stacks of posters that I have. Now I forgot whose this is. Yeah, I thought it was. Okay. All right. I will hopefully be back in 10 minutes. Email me your poster if All right, looks like the printing was a success, but so if you haven't emailed it to me, you can still do it and I'll print it out. So I printed out, I think, everything that I had received, but if I missed something or somebody, just let me know and we'll print them before 1:30. All right, let's keep looking at some more polymerization. And so now we're kind of looking at these cross-linkage reactions, which are sometimes like considered kind of polymers, or like they're not a true polymerization reaction, but they can still do like polymer-ish reactions. So we'll go over another one that's similar but different. And so these are kind of, again, reactions that you may be familiar with. So is anybody heard of that like hair thing called like Olaplex or something? Yeah, and I don't know if like people use it. And so it turns out that this does have like actual chemistry behind it. And so that's why I think it's pretty cool. And so how the Olaplex works is that it works to repair the disulfide bonds. And so that's what causes kind of hair damage, is that these bonds get broken. And so this Olaplex works to just kind of repair that. And so again, this is like a cross-linkage reaction that kind of helps reconnect polymers. So not a true polymerization, but still can, again, make larger structures. So I guess that's where we're going, right? Is that it takes our smaller structures and then kind of reconnects them. And so kind of another reaction, this is another fun one to do kind of as a good outreach demo. And so it's not often used, but the compound is sodium silicate. Yeah, so if, I don't know if you do pottery then? Oh, okay, nice. Yeah, because usually you have to go to like some pottery shop or something. Otherwise, I mean, now you can order everything off of Amazon, I guess. Plus some sort of alcohol. And so I think ethanol kind of works the best, at least when I used to do this. You could also do like isopropanol or some simple alcohol. And so and essentially, right, the same sort of thing happens. It's a cross linkage reaction. So you have all these, right, silicates, and then your ethanol can come in, right, and kind of make these cross linkages, and then you can make, right, other cross linkage reactions, so that essentially, right, you start getting this silicon oxygen bonds forming. And so that's how you kind of make this, we'll call it a cross-linkage polymer. And this is pretty fun to do, like I said. So you kind of mix the two chemicals together, and then it starts solidifying and forming this like goopy ball thing. And it probably gets rid of water, from what I suspect is a product. So it's probably a condensation reaction. And so you just kind of like squeeze it together, and it makes this nice little somewhat bouncy ball. I mean, eventually it does like crumble, since, like I said, it's not a true polymer, but it still does make a nice little rubber ball to kind of play with for a while. So those are your cross-linkage reactions. All right, let's see how far we get in our proteins, and then we're starting to touch into like amino acids and eventually DNA. And so of course we can consider a protein is essentially a polymer. And so now a protein is a biological protein whose monomer unit is an amino acid. And I should have printed out your cheat sheet, so I'll do that probably for Thursday, have this printed out. And so here are all our amino acids. And so this is just the rough structure, right, of an amino acid. And then so go ahead and identify the different parts. And so usually this is a chiral center, which we've looked at that paper in here, right? The chirality of life. Yes, no? Yeah, yeah, I feel like we have. So if I need to pull that paper up again, we can. But at least we're just kind of thinking about maybe how life started, or, I mean, if you want to go there. But, okay, key point being is there's a chiral center, so all life has a certain handedness, so a certain chirality. And then you can go ahead and kind of star glycine in your little chart here, essentially. So glycine, if you look at it, it's unique. It's the only one that's not a chiral center since it has a hydrogen on this carbon here. So instead of an R group, right, it has two hydrogens. And maybe let me redraw it in this format. And so that's glycine. And so glycine is the only one with no chiral center. But the rest all have, and again, right, definition of a chiral center for our carbon is that it has 4 different things attached to it. And so every amino acid is going to have a hydrogen, some sort of R group, right, that makes it unique. And then they all have an amine and a carboxylic acid group. Okay, and then maybe already mentally you're starting to think of how they might chain and pair, right? So here we go again. We have an amine, we have a carboxylic acid, so we could probably form an amide bond again, just so we're starting to see how these monomers are going to link back up. So let's just kind of look at our amino acids. So I like this chart that I have pulled up just because it gives us kind of a good rough guide to things. And so just kind of pointing out a few notes. So we have aromatic, right? So anything that has our aromatic ring on there. And so phenylalanine, tryptophan, right, tyrosine. So those are, again, just ones that have rings on them. And then tryptophan is usually like a good drug precursor, or there are drugs that are similar in structure to tryptophan. Sorry, printer emergency. People are trying to figure out the printing. And then aliphatic is just not aromatic, right? So you can see most of them are aliphatic. I mean, you could start to get into like acidity too, and then that gets into like reactions under different pH conditions. So you can just kind of maybe see who's susceptible to attack as well, right? So aspartic acid, for example, is really prone. So we have that, right? Another carboxylic acid group there. Same with glutamic acid, right? So again, very pH dependent. Other ones, so sulfur-containing ones. So sulfur could sometimes be like susceptible to attack, for instance. So like silver can disrupt these sulfur bonds, and so that's how like antimicrobial stuff works. So the sulfur is susceptible, and then we have right our essential amino acids, and so for example, I know my cat needs D-methionine in his food. I forget what it does exactly for the cats. I think it helps something with like urine blockage, but I actually don't know where it comes from. But yeah, so oh. Yeah, it's I assume it's this because I did a lot of research to like find the stupid cat food that has that. So my cats can survive. So let's see. Just trying to point out, yeah, valine, proline, all these are pretty common. Leucine, isoleucine. Then again, you can tell which ones are essential. And so it states up here that essential amino acids must be obtained from the diet. So we won't go there today, but right, if you were taking like a nutrition course or were interested in like nutrition, you can look into all the amino acids and where they're sourced from. Unfortunately, I'm not much of a biochemist, so I don't know off the top of my head. And so like we mentioned kind of before, so all of these are chiral centers. So we could have L-alanine and D-alanine. And I think life is L-based, but don't quote me. And if you had, right, a racemic mixture, that's when you have a 50-50, right, DL mixture. So, right, that means that you have both of these in the same quantity. So it depends, right, how it's synthesized. The other kind of interesting thing, and this is kind of interesting if you're interested in being, right, some sort of peptide protein chemist or something, but I've heard stories that scientists that work with this, like literally work with the powders on a daily basis, eventually start to develop some sort of allergy to some of these, the synthetic amino acids. So, not sure. Something to look into further. And so then we can also get into describing our proteins. And so our protein structure, kind of the first level, is the primary structure, which is just our sequence of amino acids. So not getting into, right, the folding or anything like that. So just, okay, what's our sequence? What order does it go in? And I mean, there's all sorts of research too in this area. Let's see. Should have been faster to pull up studies. But basically, you can do like essentially these like labeling experiments, and then you have this, it's like literally a little bead, and then you can, you know, put your peptide on there. So I'm just kind of making up whatever peptide. And so kind of the other thing is like people do what's called these screening tests. And so if you have to do it by hand, sorry, I'm trying to pull up a paper. But basically the idea is that you can make like a library of different peptide sequences. And so you could even think, right, maybe you have a simple one that's like 6 or 7 amino acids. And then you want to think about like, okay, well, how do I get the maximum variability? And so you might start off with like 7 tubes and you attach, you know, 1 amino acid. Then you combine them, mix them, redistribute them into 7 new tubes, and then like do it again, right, to get all the permutations of the amino acids. Does that make sense? Sorry, not doing a good job finding these papers. But what they're used for, at least like some of the hot areas of research, if I can ever pull up a paper, is they were used for like cancer detection, so trying to do like early cancer screening. And then the idea is, okay, well, you develop, in this case, the particular example I was thinking of was they were trying to develop this triligand peptide, but here, we'll pull up some other papers that are similar. Okay, well, obviously I can't find the paper I'm thinking of. Well, here's another one. It wasn't the exact one I was thinking of. So this one, they have some sort of like tertiary protein, but apparently they're trying to screen for like inhibitor of a botulism neurotoxin. So that's kind of interesting. But yeah, now I can't find. They must have older papers somewhere else. But yeah, basically they were using it for like cancer screening as other things. So right, you're just trying to find some sort of peptide that is sensitive to whatever marker you're interested in, right? So you would screen for this marker, and then if it hits it, the bead, at least the system that I was familiar with, would like light up purple, and then you like literally pick out the bead, and then put the peptide, get the peptide synthesis or sequence, excuse me, so that you would know what the successful peptide was. And then, like I said, the whole point was try to develop early cancer screening. And so then we can see again how these peptides come together. And so again, it's kind of a condensation reaction. Although I will say in practice, peptide synthesis, if you're doing it by hand, so kind of what I was saying is like people do this by hand, but at least if your group is rich enough, maybe you get an automated machine, and then the machine starts pumping out all your peptide sequences that you can screen against, and then that's a lot easier to deal with. Otherwise, as a safety hazard, the synthesis uses piperidine, which is a reproductive toxin. So I don't recommend going into that. No, I'm joking. But yeah, that's one of the nastier ones. But anyway, so like we're looking at, right, similar mechanism to all these condensation reactions. And maybe we'll put that again. Okay, this is another condensation reaction. So once again, we have our amine, our carboxylic acid, right, they're coming together. Same mechanism, right, nitrogen's attacking, blah, blah, blah. Water leaves, amide bond formation. And so this is the key thing that you're looking for is, okay, where's my amide bond? Because again, right, if you have to, well, let's say now we want to draw out an alanine serine dipeptide, right? So just two amino acids. So if it's alanine first, we start with our amine. So right, this amine's intact. Then amide bond formation, right, between the carboxylic acid group of our alanine on the end here, and then connecting it to serine on the nitrogen end. Does that make sense? So kind of take that in for a second. Oh, let me get this out of the way. And so that's kind of my suggestion, as you kind of see, and maybe this is like to your point of what's the backbone, but then I would ask you to kind of point out, okay, where's your backbone, right? And so there's your alanine, so amine, carboxylic acid, and R group, right? And so you always have this carbon, carbon, and then, right, amide bond. So amide bond, then carbon, carboxylic acid again, just kind of repeating. So there's your R group, right? So you can identify your amino acid. Now here's serine, right? Again, that's that R group. So we could go back to our cheat sheet that I forgot to print out for y'all, and then find, right, serine here. And then, okay, that's the group hanging off of it. And they drew it kind of, I would say, a little bit different looking, right, than kind of this guide. But I like the flat projection when we're just trying to, like, chain these things together. Because I'll admit, I go a little loopy, too, trying to see, like, where they bond from. So, again, just taking that in. So, serine, right, this is the R group. So, that's what makes it unique. So going back, okay, so that's what's down here. And then just to kind of compare, well, what if we did serine alanine next? And so right order is flipped, and so that means it's just slightly different. Maybe let's zoom out so we can see both of those at the same time. And now serine's first, right? So amine, the R group, amide bond formation again between the two, and maybe, right, kind of notate where one amino acid is, and then the second one begins. Again, we're losing water. So just kind of take that in, since like I said, on the exam, you'll probably be asked to examine an amino acid sequence and see if it's correct or not. And so for that, this is like what I would call our cheat sheet template then, right? So we have amine, our carbon, carboxylic acid, amide bond. And so just kind of look for that R group on our carbon, amide bond again, right? And kind of highlight like, okay, that's one amino acid. Here's our next amino acid. Then our third one, right? Just keep kind of pointing out those amide bonds that separate your amino acids. And so just on and on and on. So again, use this template to kind of chain them together, and then your only variability is in the R group that makes your amino acid unique. All right, so like we mentioned, we have our primary structure. And then, of course, some of these primary structures get pretty long already. And so we can, again, just kind of appreciate this information. So insulin, right, that's now made synthetically. And so that is shorter at only 51 amino acids. And then different toxins, so snake toxins, keratin, right? Again, you're just kind of appreciating the variability in size too. So notice. Uh, sorry, I think I didn't understand because it is made up of like 4 polypeptide chains. So I think this should be like for all of them, if that's what you're asking roughly. Yeah, so I think that's what it's saying by this count. Because this is, but yeah, I mean, like you're saying, this is only the primary structure though, so it's not giving us any of that. And then, so like you were saying, then we would have to have, right, more information of how then these primary structures actually fold. So let's look at some pictures. And again, these are just kind of very basic. So obviously, this is not all inclusive. Oh, come on. Where's a good picture? I feel like Wikipedia has some of the best pictures. And so kind of the main structures are the alpha helix, which is this kind of coiled structure, versus the beta sheets, which are kind of these long strands. And so again, that's just kind of looking at our initial folding. And there are other shapes. Don't know if Wikipedia is going to have them all. And then I was trying to get a good picture of hemoglobin since you brought it up. So yeah, so I think this count was for all of them. And then like you were saying, it's, well, it has it shown here. So it's 2 alpha and 2 beta helixes are kind of the main structure there. And so trying to show it so the alpha is in red and the beta's in blue with the folding. So again, just kind of taking it in for now. And then the tertiary structure is then the full kind of 3D shape of our protein. So this is, right, kind of our folding then and extra interactions. And interactions. And so probably like some hydrogen bonding interactions as well, right? How they fold and interact. And we've looked at how like some diseases are caused, right, from misfolding, and that could be in either the secondary structure. I think we looked at that where something was like the wrong structure, or sometimes that can also happen with the folding. And then I was trying to see if I put it on my notes here. So then I neglected to put on my template, but we should at least put quaternary structure, which is then the interactions between two proteins. So just seeing how then, right, the proteins start to interact with each other. All right, we probably, even though it's like a little early, we might want to stop there before we start our new chapter. And so we can see kind of where we're headed on the last bit of Thursday's lecture. So Thursday we'll do the nylon demo as well, but then we'll finish up with our last kind of example of polymers. So we'll be looking at DNA or polynucleotides, right? So kind of looking at how DNA is a polymer, which we did tease right at the beginning of today's lecture, but we'll finish up on Thursday looking at DNA, and then of course how it forms, right, the double helix, which is pretty interesting. But I think we should end just a little early to give people time to finish last minute printing and posting. And so we can also use like the ring stands in lab. Yeah, we'll go over in lab how we'll do it, but we'll probably divide into kind of 3 groups roughly. And so then, you know, you'll be, if you're in the group, you'll be presenting, and then everybody else can be audience and kind of wandering around, looking at the posters, and we'll run it as a poster session. Any other questions? So make sure you get the homework. So there's homework, right? We're done with labs. We just have to do our poster presentation. Oh, and then papers are just due by the end of the quarter. So I'll just put that on here. So papers by the end of the quarter. Yeah, email would be preferred, or paper, hard copy is also preferred. But yeah, email's fine. And I'll accept them probably up until like Sunday or whatever. Any other questions or anything? And so we have tape. We should have glue. I can grab more tape if we need. Email me if you haven't already. I think I've got most people's printed. I was going to go check my email and print out other things. And yeah, we can kind of start migrating to lab as well. And then a few people have kind of dipped out at the last minute, so we will do like a Thursday mini session. So yeah, make sure Thursday. But snacks are today, so I can't promise snacks for Thursday. Yeah. All right. Any questions on stream? I'll leave it going for a second or two. And then again, just get stuff printed. We'll start meeting in lab.